All right, so let's dive into more of Chapter 8 and talk about some chromosome mutations. There's three major kinds: a rearrangement, aneuploid, and polyploid. Chromosome rearrangements can come in a couple of different varieties, but all of them have to do with the actual structure of the chromosome being altered in some form. Aneuploid mutations are where you are actually seeing a difference in the number of chromosomes that are present. So an alteration, whether it's deletion or or addition. And then polyploids is where complete sets of chromosomes are going to be added. So this becomes really important when we're talking about hybridization of some of our crop species. So the four chromosomal rearrangements that we're going to be talking about are duplication and deletion, as well as inversion and translocation. So a chromosomal rearrangement is going to be a mutation that is going to change the structure of an individual chromosome somehow. So many of these are going to occur when double-stranded DNA actually breaks. So the DNA strand actually breaks, and this happens fairly often. But the system has, or the organism has actually developed ways to deal with that and try to repair itself, and that's where these rearrangements kind of come from. So what ends up happening on a pretty regular basis is if we get double-stranded DNA that is breaking, a lot of times if it doesn't rearrange itself correctly or in a functional manner, then the cell ends up dying. So the mechanisms that you see here on this slide are just some of the rearrangements that have evolved through evolutionary process to help reconnect the broken ends of these DNA gaps that have broke. So some of the things that you can kind of run into is if you have a wrong end of DNA that is actually connected, then you get some sort of rearrangement. And so this can happen four different ways, like you see here on this slide. But you can also alter the way that that crossing over can happen. And so incomplete crossing over or inappropriate crossing over can actually result in some of these rearrangements as well. So let's first start talking about duplications. There's two main types of duplication: tandem and displaced. Tandem duplication is where you are going to have you're going to have genes that are repeated right after the original set of genes. So for example, in a normal phenotype, if we had the A gene, the B gene, the centromere, C gene, D gene, E gene, F gene, and G gene, in the case of tandem duplication, we would see the E and F gene being duplicated again and then stuck in right after the two genes that they were synthesized from. So this is what we call tandem duplication because it, just like a tandem bike, it happens right after one another. So as you can see, this figure here really relates to tandem duplication, where we have the genes E and F being repeated with duplication. And this presents a problem because as you are trying to align chromosomes in prophase 1 of meiosis, E and F on the original chromosome is only going to have one copy of E and one copy of F. But the duplicated chromosome has two copies of E and two copies of F. Therefore, to get the first copy of F to line up correctly and the copy of G to line up correctly, you actually have to, the chromosome forms this loop. And that can cause problems when synthesizing DNA as well as going through the anaphase process where these chromatids are going to have to separate from one another. The second type of duplication is going to be displaced duplication, and this is where you actually get duplication of a gene occurring. It just happens downstream, so it's not necessarily right after the gene that's being duplicated. So it could be a long distance. So for example, E and F are our original genes in question. If they were duplicated but did not occur right directly after, like in a tandem situation, then it would be called displaced duplication. And then you get into a real mess when you try to get chromosomes to line up with one another. So duplication ends up being a problem for a couple of reasons. First of all, like we talked about originally, individual homozygous chromosomes that have duplication as a part of their structure is going to carry that duplication on both chromosomes. So homozygous chromosomes are going to have duplication on both chromosomes. Therefore, matching up during the prophase stage of meiosis really isn't a big issue. But if you get into a heterozygous situation where one chromosome has the duplication and one does not, you can get into some situations where these loops would actually have to form, and you wouldn't get really good lining up of those chromosomes during metaphase and then separating during anaphase. So duplications are going to be a problem because the pairings may not be able to take place in the way that they need to because of this mismatch between the homologs. Duplications can also be a problem because we can actually see double expression and an abnormal phenotype being expressed. An example of a duplication issue that we see with flies is in the bar region of the X chromosome on flies. So here, the bar mutation has the ability to reduce the number of facets in the eye. So if you get a wild type individual that is normal, they have one bar region on each X chromosome. Okay, but if you get a mutant in a heterozygous form, then you're going to get two bar regions on one chromosome because that bar is going to be duplicated in a tandem fashion and only one on the original chromosome. So the more bar duplications that you have, the smaller the eye is going to be because it's actually tying up the proteins that create the facets in the eye. And then if you get a homozygous situation to where you have duplication on both chromosomes, again, those proteins are going to interact with each other and you're not going to get facet creation, so the eye is going to be even smaller than if it was in a heterozygous fashion. And then in the heterozygous fashion, another heterozygous phenotype or genotype that you can see is a double bar, where you end up with 3 bars on one X chromosome because of double tandem duplication. And so this really reduces the number of facets in the eye. So those mutant flies tend to actually be lost as part of the larva process and don't really get to live for a very long time. So when we look more closely at this bar interaction, the duplications and deletions are actually going to occur pretty often because of unequal crossing over. So during prophase and meiosis, you're going to see a lot of crossing over and a lot of chromosomal information exchanging places. So if that doesn't happen in equal proportion, then you get these extra regions or these duplications within those chromosomes. So the duplicated segments of the chromosomes then are not going to be able to align with each other the way that they need to in metaphase, and you're going to get that loop fashion that we saw a couple of slides ago, and that's really going to alter the way those chromosomes match up and as well as separate in anaphase. So this this characteristic right here, that even though we're looking at it as far as bar mutations in flies, this is actually really the premise and the cause of the red-green color blindness that we see in humans. It all has to do with the amount of red-green facets in our eyes and the amount of protein that is synthesized in the absence or the presence of those genes. All right, so how does duplication alter the phenotype? And this gets a little bit convoluted because, and it's a little less straightforward than like a deletion, because genetic information with duplication is not missing. There's just extra copies of it. So you have more information than you actually need. So when you get additional copies of the same gene, you get abnormal amounts of gene products or protein. So the gene is going to go through transcription, where we're going to get synthesis of messenger RNA, and then that messenger RNA is going to go through translation, where we actually are building amino acid blocks of protein, and it's the protein that is going to be expressed. So if you have more than one gene present, more than one set of gene that is being transcribed to mRNA and then to protein, the gene dosage is going to be off. So if you end up with two copies of a normal genotype, that's going to produce the one or the amount of protein that is normal. However, if you get three copies of a gene, then you're going to actually produce 1.5 times the protein that is normal. So you're going to get altered phenotypes. So we can actually see this type of relationship in bass. So if you end up with a chromosome of a wild type individual where they have genes A, B, and C on this chromosome, in normal situations, that's going to go through transcription and translation and produce these three gene products. When those three gene products are going to interact together, and they are going to cause this growing embryo to display the correct or the normal development that we see in these bass species. So in an altered form where we see duplication taking place, for example, if we have two copies of the B gene, then we have two copies of B protein that are interacting, and they are going to cause an alternative phenotype to occur in this growing embryo, so you get abnormal development. Then if the amount of one product increases, but the amount of these other genes don't increase, then you can see some developmental problems actually happening. So one example that we see with duplication is actually with globin proteins in humans. So we know that we need hemoglobin in our blood to carry oxygen throughout our body. Well, those globin proteins have evolved over time. So now we have specific globins that are going to be activated when we're adults, and we have specific globins that are going to be activated in utero as a fetus. And they have two very different jobs, but we were able to evolve because of some of the extra genes that are now associated with those globin molecules.